नोबेल प्राइज इन मेडिसिन किसे मिला है? नोबेल प्राइज इन मेडिसिन हैज गॉन टू विक्टर एम्ब्रोस एंड गैरी रोफकुन फॉर द डिस्कवरी ऑफ माइक्रो आरएनए. व्हाट इज माइक्रो आरएनए? व्हाट इज द पर्पस ऑफ माइक्रो आरएनए? अच्छा, बिफोर वी अंडरस्टैंड माइक्रो आरएनए, क्योंकि माइक्रो आरएनए में प्रोटीन सिंथेसिस के बारे में थोड़ी सी बात होगी, डू वी अंडरस्टैंड हाउ प्रोटीन इज फॉर्म्ड इन आवर बॉडी? Do we all understand how protein is formed in our body? देखो, we have cells. Within the cell, we have nucleus. Nucleus में we have DNA. Some DNA we got from our father, some DNA we got from our mother. Some chromosomes we got from our father, some chromosomes we got from our mother. And उसने हमें एक unique DNA दिया है. And that unique. Do I need to do cover vision PT 360 have prelims? नहीं, you do not have to. मैंने बोला ना, it is my responsibility to ensure that everything that you need for this exam. देखो, कोई बच्चा जो हमसे पास्ट में पढ़ा है ना, उससे पूछना. They will be the right guide rather than मैं बोल रहा हूँ. They will tell you this कि इतना पढ़ के चले जाओगे वो बहुत होगा. It's my responsibility to go through all the material and to ensure that कोई भी चीज जिसमें relevant है वो आपके नोट्स में रहेगी. आपको मैं ना एक वीडियो बना नहीं पाया हूँ, मोस्ट प्रोबेबली मैं कल बनाऊंगा. इनफैक्ट कल अगर समय रहा तो मैं लाइव क्लास में ही करता हूँ. ये वो वीडियो था इस बार मेंस एग्जामिनेशन में कितने क्वेश्चंस करंट अफेयर्स प्रोग्राम से है. अब सब बोल देते हैं देखो मैंने ये टर्म पढ़ाया था, ये पढ़ाए थे. वैसे नहीं, एग्जैक्टली जो पढ़ाया है उससे कितने क्वेश्चंस है. वी हैड 17 क्वेश्चंस फ्रॉम करंट अफेयर्स प्रोग्राम. स्टूडेंट्स वर वरीड अबाउट कि टारगेट ऑफ सेंडाई फ्रेमवर्क. हमने टारगेट ऑफ सेंडाई, 7 टारगेट एकदम बच्चों को रटवाए थे क्लास में. क्यों रटवाए थे? क्योंकि सेंडाई फ्रेमवर्क पे सवाल आने का चांसेस हमेशा रहा है और बहुत इजी था. हमारे बैच में पढ़ा हुआ था. इस बार मेंस एग्जामिनेशन में उड़ान स्कीम पे क्वेश्चन आया है. माय पॉइंट इज कि वी ट्राई टू इंश्योर दैट ईच एंड एवरीथिंग व्हिच इज रिलेवेंट फॉर एग्जाम वुड बी टॉट टू यू. एंड किसी और सोर्स को पढ़ने की जरूरत नहीं है. विल मेक श्योर दैट एवरीथिंग इज देयर इन योर नोट्स. इंश्योर, मुझे एक प्रॉमिस चाहिए कि आपको जो पढ़ाया गया वो आता है. आई कैन प्रॉमिस यू दैट विल बी इनफ फॉर योर सिलेक्शन. अगर आप प्रिलिमिनरी एग्जामिनेशन की बात करते हैं, अगर आप प्रिलिमिनरी एग्जामिनेशन में जो आपको टेस्ट में असाइनमेंट मिल रहा है, स्टैटिक पोर्शन आपको खुद से पढ़ना होगा. हिस्ट्री, ज्योग्राफी, पॉलिटी, इकोनॉमी का बेसिक्स वो आपको असाइनमेंट के फॉर्म में मिलेगा. अगले संडे के टेस्ट में यू विल गेट हिस्ट्री एंड पॉलिटी एंड इकोनॉमी पोर्शन ऑल्सो, स्टैटिक वाला जो आपको खुद से पढ़ना है. एंड विल हैव करंट अफेयर्स पोर्शन ऑल्सो. In this way, we'll go ahead to if you keep revising, cover the static portion basic and follow the current affairs properly, you will have enough marks in your preliminary examination, enough marks in your mains examination. Will there be discount for those who have once registered for any of the course last year? देखो, अगर जो करंट अफेयर्स वाले बच्चे हैं लास्ट ईयर वाले, उनके लिए तो कुछ डिस्काउंट है, बट उसके लिए आपको ऑफिस में कॉल करना पड़ेगा. लेट्स कम टू प्रोटीन वाला बात. हम प्रोटीन फॉर्मेशन की बात करते हैं. देखो, how is protein formed in our body? तो we have cell के अंदर nucleus है. Now let me make a bigger cell. Let me say this is the cell and this is the nucleus. Inside the nucleus we have DNA and on DNA we have genes. Those genes are basically encoding for protein. Those genes are basically encoding for protein. They have coded for a protein. So which protein will be formed in my body will be decided by what gene I have. But let me draw this DNA. DNA के ऊपर gene होता है. We have 20-25,000 genes and thus we can have 20-25,000 different protein. Now protein is manufactured outside the cell in protein factory called ribosomes. Protein is manufactured outside the cell by protein factory called ribosomes. They are not manufactured inside the cell. Where is the information given of which protein has to be formed? Inside the cell. Genes don't come out. तो यहाँ पे क्या होता है? Genes से एक messenger RNA बनता है. And this messenger RNA goes out. It goes out and tells ribosomes this protein has to be formed. Okay, abhi we will when we study biotechnology from basics. January में हम science and technology पूरा पढ़ना शुरू करेंगे, एकदम static सब कुछ पढ़ते हुए. Like फॉर सम सब्जेक्ट्स मैंने आपको बोला है आपको हिस्ट्री, ज्योग्राफी, पॉलिटी, इकोनॉमी खुद से बेसिक्स उसका खुद से पढ़ना है। हिस्ट्री, ज्योग्राफी, पॉलिटी, इकोनॉमी का मैं आपको करंट अफेयर्स पढ़ाऊंगा खाली इन करंट अफेयर्स प्रोग्राम। बट फॉर सब्जेक्ट्स लाइक साइंस, एनवायरमेंट, इंटरनेशनल रिलेशंस, इंटरनेशनल बॉडीज, मैपिंग, आपको कुछ नहीं पढ़ना है। इसका मैं सब कुछ पढ़ाऊंगा आपको, इवन द बेसिक पोर्शन भी पढ़ाएंगे। ओके, तो लेट्स कम बैक। व्हाट हाउ इज प्रोटीन फॉर्म्ड? हमारे सेल के अंदर न्यूक्लियस है, न्यूक्लियस में डीएनए है विच वी हैव गॉट फ्रॉम आवर पेरेंट्स, and on those DNA, parts of DNA, sub parts of DNA are known as genes. Those genes encode for protein. Those genes have code for protein. Protein कैसे बनता है? Protein is basically a chain of amino acids. तो amino acid कौन कौन से जोड़ के, which are the amino acids which have to be brought together to form a protein in your body? Protein which is formed in my body is different from protein formed in your body. देखो मेरा स्किन कलर आपके स्किन कलर, मेरा हेयर कलर आपके हेयर कलर, हम सबका यूनिक है because different kind of protein is being formed in our body. Okay, so my question was कि how is protein formed? Protein is formed with the help of messenger RNA. तो जीन से, डीएनए से जब ये आरएनए बना है, इस प्रोसेस को हम बोलते हैं ट्रांसक्रिप्शन। फिर आरएनए से जाके प्रोटीन बनेगा। इतना तो समझ में आया हमें? आर वी ऑल क्लियर अबाउट इट? ओके, नाउ आई हैव अनदर क्वेश्चन. Question is, do all our cells in our body, there are billions of cells, do all our cells have the same DNA? except let's say red blood cells जिसमें न्यूक्लियस होता ही नहीं है, बट जनरली क्या होगा, हमारे सारे सेल्स में सेम डीएनए है. व्हाई? बिकॉज when you think of how our life began, it began as a single cell. Mother's egg fertilized by father's sperm, वो अपने क्रोमोसोम्स लेके आए, ये अपने क्रोमोसोम्स, स्पर्म में क्रोमोसोम्स आए, फर्टिलाइजेशन हुआ, एक यूनिक सेल बन गया, फर्टिलाइज सेल, जिसे हम जाइगोट बोलते हैं. आफ्टर दिस व्हाट हैपेंस? इट इज दिस सेल ओनली व्हिच कीप्स ऑन मल्टीप्लाइंग, so it will all have the same nucleus and thus the same DNA, right? So every cell in our body has the same DNA. Within the nucleus has the same DNA. Are we clear about this? It has the same genes. Are we clear about this? Fine. But do every cell in our body produce the same protein? No. Think of this, the cells which are here, आपके हेयर फॉलिकल्स में, वो तो ये बाल बना रहे हैं, बट द सेल्स which are in your heart, are they creating these hairs, protein for these hairs? नहीं ना. अगर वैसा होता तो जब दिल निकाला जाता बाल बाल वाला दिल आता, राइट? एवरी सेल is not making the same protein. Are we clear about it? Every cell is not. Our pancreas are making different protein, our heart is making different protein, our heart cells are making different protein, our skin cells are making different protein, our follicles are making different protein, right? So not every, but उन सब में DNA same है. Take out any cell from your body and you will find exactly the same DNA which was there when the fertilization had taken place. जब ये single cell, ये egg cell, all the life on earth starts with a single cell. Even all of us now today we have billions of cells in our body, but we began as a single cell. Mother's egg cell was fertilized by father's sperm to form a zygote. This is how we started, and this zygote only has multiplied to create this complex human being. So all the cells in our body have the same DNA. So let's say this is the DNA which is there in all the cells of our body. Now parts of DNA are known as gene and they are encoding for protein, but not all the proteins are getting expressed in every cell. इतना तो समझ में आ गया? Not all the proteins are expressed in every cell. Our pancreas में जो cells है, they are expressing some other protein. So they have gene 1, they have gene 2, ऐसे ही they may have 20,000 different genes, 20,000 plus different genes, but not all genes are getting expressed. Not all genes are getting expressed in every cell. In different cells, different genes are getting. हमारे in our body are all cell types the same? No. Our muscle cells are different, our heart cells are different, our pancreas में जो cells है different है, our neuron cells are different, our skin cells are different. सारे cells भी same नहीं है. The protein which they are manufacturing is also not same. What is same? Their DNA. उनके न्यूक्लियस के अंदर जो DNA है, that is same. But they are functioning differently. How this regulation happens is the subject of this year's Nobel Prize in Medicine. Do we get it? Do we understand it? So, if you'll come here, basic question was, our organs and tissues consist of many different cell types, all with identical genetic information stored in their DNA. However, these different cells express unique set of proteins. They are not expressing the same set of protein. देखो, different cells हमारे brain में जो cells हैं, cell type, it is creating, expressing some other protein. Muscle cells are expressing some other proteins. So, cells are same, genetic material is same, but the proteins which are being created by them are unique, are different. Not all proteins are different. Some proteins may be same, but different types of cells are also creating different kinds of proteins. How does that happen? Okay, this is the problem that the current scientist solved. पहले problem समझना बहुत जरूरी है. Only then we'll be able to understand how they have solved it, and only then we'll be able to understand what is the significance. पहले the things that we are studying right now, the things that we are trying to understand right now, that will give us very fundamental basic understanding, so that no matter what question gets asked in the examination, we'll be able to solve it easily for preliminary examination. But आखिरी में हम पढ़ेंगे 5-6 significance of this that will be relevant for our mains examination. इसका application क्या है? Because mains examination में questions कैसे आते हैं? Application, significance वाले. Whereas preliminary examination में they'll ask you anything. RNA interference technology से इनमें से क्या हो जाएगा? या microRNA contributes in which of the following? और तो you have to know microRNA from fundamentals to actually able to recognize ये नहीं हो सकता, ये हो सकता है. Because they may give you anything, whereas there they'll ask you give me applications, and you will be able to write your own applications of microRNA in the mains examination. Okay, we understand the problem. Now, before discovery of microRNA, when scientists were asked कि यार ऐसा क्यों हो रहा है, like how is it happening that some cells are producing some kind of protein, other cells are producing other kind of protein? How does that work? So, in 1960s, the concept of transcription factors was used. It was said that it is this transcription factor which are responsible for some cell expressing some protein, other cell expressing other protein. So, what are these transcription factors? These are specialized proteins found in our body, and they bind to specific region in DNA. So, basically, यहाँ पे image नहीं है. Let me come here. This is my DNA. We are saying this transcription factors are basically proteins which will come and bind on our DNA. By this, they will control whether this gene will be expressed more or this gene will be expressed less. So, what are transcription factors? Pehli cheez, if you are asked what is transcription factors are protein which controls whether a gene will be expressed more or a gene will be expressed less. In our heart cells, बाल बनाने वाले प्रोटीन का gene will not be expressed. तो वहां पे transcription factor, it can bind to specific region in DNA. So our transcription factor binds to the specific region in DNA and control the flow of genetic information by determining which mRNA are produced. जीन से क्या बनता है? mRNA बनता है, तो it will control which mRNA is getting produced, and thus is controlling which protein is getting produced. Because जिस RNA का, जिस DNA का mRNA बनेगा, mRNA only is taking information to the ribosomes to make this protein, manufacture this protein. तो जिस जीन का mRNA बनेगा वो ही, only that gene is going to get expressed. So this factor, transcription factor, controls which mRNA will be formed. Okay, now transcription factors can be of two types. One is activator, other is repressor. Let's say this is my DNA and इस जीन का transcription factor आके यहाँ पे attach हो रहा है. If this transcription factor is activator, it will lead to more formation of mRNA and thus this gene getting expressed more. अगर ये transcription factor activator है, it will boost the production of mRNA and thus boost the production of protein. Whereas if this transcription factor is a repressor, it will resist or decrease production of mRNA and thus decrease the production of protein. ये बात समझ में आई? Did we understand the meaning of transcription factor? Are we all comfortable with the term transcription factor? So what is transcription factor? Transcription factors are proteins which get attached to DNA and restrict or promote formation of mRNA. वो जिस जीन के DNA के जिस component पे attach होंगे उसमें they can work as activator or they can work as repressor. So in 1960s when it was understood कि why a cell is forming this protein, manufacturing this protein, whereas the other cell is manufacturing other protein, our understanding was on the basis of transcription factors. It is transcription factors which are controlling everything, because at that time we did not know about microRNA. Okay. However, in 1993, this year's Nobel laureate, this year means 2024 के जो Nobel laureates हैं, they published unexpected findings describing a new level of gene regulation. Which gene is getting expressed, which gene is not getting expressed is gene regulation, and they, by discovering microRNA, explained other mechanisms by which gene is getting regulated. Okay, how did these scientists contribute? Let us understand. अभी तक हमने background ही समझा है. Before microRNA was discovered, what was considered the thing which controlled which gene is getting expressed, which gene is not getting expressed? Transcription factors. So do transcription factor really exist? So yes, they exist. Do they regulate gene expression? Yes, they regulate gene expression. They also function, but what also functions? MicroRNA. In 1993, they, for the first time in a research paper, published this year's Nobel laureate published about microRNA. Basically, they were working on C. elegans. It's a roundworm, a very small roundworm. They were working on it, but this roundworm, though very small, produces various kinds of cells. इसके पास muscle cells हैं, neuron cells हैं. So through the study of this development of this roundworm, they were trying to understand when does a cell, we were initially what? A single egg cell. It has specialized into muscle cell, neuron cell by producing different proteins. So how does this differentiation occur and what is responsible for formation of this protein? Before this, what was the reason given? Transcription factor is the reason. But the two scientists were interested in genes that control the timing of activation of different genetic program. That is, when different genetic programs start getting expressed in our body. Initially, egg cell mein same DNA, same protein, but it specializes into different kinds of cells as multiplication happens. So how that is happening, they were trying to understand that. And it was Victor Ambrose who in his lab at Harvard discovered microRNA, a short RNA molecule that lagged code for protein production. This microRNA will do what? Come here. He discovered microRNA. This microRNA will not prevent the formation of mRNA. Transcription factor was doing what? Transcription factor was preventing or promoting the formation of mRNA. It did not do that. ये microRNA, he found, after mRNA has been formed, it blocks the functioning of mRNA, and by that blocks some gene from getting expressed. So in my heart, the gene related to protein which will create hairs are not getting expressed. They are not getting expressed. Why? Because some microRNA is blocking the mRNA of this mRNA of the protein which creates my hair. Are we getting it? So he found this for what? He found this for roundworm. In his lab at Harvard, discovered microRNA, a short RNA molecule that lacked code for protein production. So this RNA molecule itself did not produce protein, but Gary Rivkin found that microRNA did not inhibit the production of mRNA. microRNA is not inhibiting the production of mRNA. mRNA is getting formed, but the regulation appeared to occur at a later stage in the process of gene expression through the shutdown of protein production. So the microRNA is not preventing the formation of mRNA. The regulation is happening after that, after mRNA has been formed. That is what the other scientist Gary Rivkin found. Then two scientists came together, collaborated. Initially also they were collaborating in their postdoctorate period in 1980s. Then they moved to, after their postdoctorate, they moved to different places, but they were doing research in the same field. They collaborated and found that microRNA produced blocks that target gene by binding to complementary sequence in the gene's mRNA, which eventually blocks the production of protein. So basically, microRNA blocks attaches to mRNA, which blocks the production of protein. It is what they found. Thus, they developed or they discovered a new method of gene regulation. अभी तक gene regulation का method क्या था? Transcription factor. Here, new principle of gene regulation was found, mediated by previously unknown type of RNA, microRNA. Previously, microRNA were also not known. Here, they were able to find a new kind of RNA called microRNA. When it was published in 1993, scientific community was not very excited. 1993 में when microRNA was published for the first time, scientific community was not very excited because they thought that this was something which was a peculiarity of this roundworm. कि उस roundworm में regulation ऐसे होता है, not in other complex organisms, so it's not going to affect humans. Are we getting it? Are we understanding it? So in 1993, when microRNA was discovered and it was published, scientists were not very excited because they said their study is on roundworms, and thus the microRNA system of regulation of genes happens where? In roundworm. But the perception changed in 2000 when Rivkin's research group published their discovery of another microRNA which is found in even complex organisms including humans. And since then, more than 1,000 genes for different microRNAs in humans have been found. So, like in human body only, there are around 1,000 microRNAs which have already been found. Please understand, microRNA will be formed from what? From DNA? Please, समझो इस बात को. What we have DNA. From DNA, mRNA is formed. From DNA only, microRNA would be formed. So, since 2000, scientists in India, scientists studying human beings have found more than 1,000 genes on our DNA. जिससे क्या बनता है? microRNA, and thus more than 1,000 microRNAs have been recognized even in humans, and this becomes exciting. In 1993, when we said कि वो roundworm का peculiarity है, what? microRNA is a peculiarity of roundworm. It will regulate gene expression in roundworm. Then no one was interested. What will we do? But when we realize, okay, this is impacting gene regulation in not only roundworms but also in other complex organisms including human beings, it becomes very significant for us. It becomes very important for us. Now, further research in this field तो we understood कि perception तो change हो गया, this is going to be significant. So further research in this field, you'll see in addition to mapping of new microRNA, experiments by several research groups elucidated the mechanism of how microRNAs are produced and delivered to complementary target sequence in regulation of regulated mRNA. Basically, they're saying is they discovered mRNA. After that, research happened to understand how microRNA are actually produced and how those microRNAs are delivered to the relevant, the complementary messenger RNA, because microRNA are going to do what? They're going to block messenger RNA, so they have to be delivered. So scientists in future also found that. Now something very important, इसमें ना प्रीलिम्स में क्वेश्चन बनते हैं, या मैं तो एटलीस्ट बनाने का प्लान कर रहा. One is a single microRNA can regulate the expression of many different genes. एक single microRNA can regulate many different genes. A single microRNA is not for only one gene. It can regulate many different genes. And a single gene can also be regulated by multiple microRNAs. Are we understanding this? What I'm trying to say is कि this was gene 1, this was gene 2. Let's say this was a gene for microRNA. इससे बनता है expression से, transcription से microRNA. This microRNA 1, and let's say there is another microRNA 2. This microRNA 1 may be regulating gene 1 also, may be regulating gene 2 also, gene 7 also, gene 10 also. Right? One microRNA may regulate multiple genes. How? By restricting the function of their messenger RNA. इस जीन का messenger RNA बनेगा, उसके फंक्शन को रेगुलेट करके. तो वन माइक्रो आरएनए इज नॉट स्पेसिफिकली फॉर वन मैसेंजर आरएनए. In the same way, one gene or one messenger RNA can be blocked by multiple micro RNAs. It's not that only microRNA 1 will be blocking messenger RNA 1. Messenger RNA can also be blocked by multiple of them. इससे भी ब्लॉक हो सकता है, इससे भी ब्लॉक हो सकता है. Do we get it? Yes. Every नहीं, एवरी वही तो बताया ना, एवरी जीन के पर्सपेक्टिव से कोई इंडिविजुअल माइक्रो आरएनए होने की जरूरत नहीं है. वन माइक्रो आरएनए में वर्क फॉर मल्टीपल जीन्स. ओके, यस. नहीं नहीं नहीं, वन जीन कैन बी कंट्रोल्ड बाय मल्टीपल आरएनए. सिर्फ आरएनए माइक्रो आरएनए 1 से ही कंट्रोल नहीं होगा. ये माइक्रो आरएनए 2 से भी हो सकता है, माइक्रो आरएनए 5 से भी हो सकता है, माइक्रो आरएनए 10 से भी हो सकता है. व्हाट इज द great? What I'm trying to say is, let's say this is gene 1, this is gene 2, this is gene 3. ऐसे ही we have gene 20,000. इतने जीन से हमारी बॉडी में फॉर मेकिंग प्रोटीन. वी हैव माइक्रो आरएनएस, है ना. माइक्रो आरएनए 1 से लेट्स से माइक्रो आरएनए 1000. वी हैव मोर देन 1000 माइक्रो आरएनए, वी हैव मोर देन 20,000 जीन्स, है ना. एंड फॉर एवरी जीन वी हैव व्हाट? मैसेंजर आरएनए. यहाँ पे माइक्रो लिखना चाहिए, एम नहीं. एम रेफर्स टू माइक्रो आरएनए 1 टू माइक्रो, है ना. वी हैव मैसेंजर आरएनए, वी हैव मैसेंजर आरएनए. दिस मैसेंजर आरएनए, विल दिस मैसेंजर आरएनए बी यूनिक? हर जीन अलग प्रोटीन को एक्सप्रेस कर रहा है, तो दिस विल बी यूनिक. इट हैज मैसेंजर आरएनए 3, 1. What I'm saying is, if you have to block messenger RNA 1, it may be blocked by more than one micro RNA. Micro RNA 1 में also block it, 5 में also block it, 10 में also block it. ऐसा हो सकता है. नहीं, एक ही, not together, है ना. Any one of them, not any one of them. इनमें से कुछ होंगे जो 1 को block कर सकते हैं. Not all of them will be able to block 1, but more than one may be able to block 1. In the same way, what we are saying is, when it comes to mRNA 2, it can be blocked by multiple of them. So that is what, ये क्या हुआ? It should be noted that a single microRNA can regulate expression of many different genes. and a single gene can be blocked by, so this one mRNA may block this also, may block this also, may block this also. In the same way, mRNA is blocking, mRNA2 is getting blocked by mRNA1, it may also be getting blocked by this and this. Now do we understand? Do we understand this? Okay. So this is, इस पे ना, when I was reading this, when I was studying this, I thought एक अच्छा क्वेश्चन बनेगा. एक जब मैंने have to make MCQ, मैं ऑलरेडी लीक कर रहा हूँ. कोई बात नहीं, रिवाइज करो, मैं थोड़ा सा और मॉडिफाई करके I'll make an MCQ for next test. So now we have understood this. Now see, microRNAs used to block mRNA naturally. This process has been used to block messenger RNA in artificial mechanism also. This is known as RNA interference technology. हम किसी जीन का एक्सप्रेशन अपने आप से, like human beings artificially wants to do that. So we would want to create some compound which can block the expression of mRNA. Suppose one gene in our body is creating cancer, is leading to cancer. So we would want that उस जीन का एक्सप्रेशन बंद कर दिया जाए. That gene is creating messenger RNA. If we block this messenger RNA, will this gene's expression be blocked? So this is one of the things that we do in RNA interference technology. In 2018 या 19 there was a question in preliminary examination on RNA interference technology. उसमें हम क्या करते हैं, मैसेंजर आरएनए को इंटरफेयर करते हैं सो दैट एक्सप्रेशन नहीं हो. तो मेथड टू डेवलप मैसेंजर आरएनए इंटरफेरेंस कैन बी लर्न फ्रॉम माइक्रो आरएनए, बिकॉज व्हाट इज माइक्रो आरएनए डूइंग? माइक्रो आरएनए इज इंटरफेयरिंग विद मैसेंजर आरएनए एक्सप्रेशन. तो cellular machinery for producing functional microRNA is also employed to produce other small RNA molecules in both plants and animals. For example, as a means of protecting plants against virus infection, Andrew J. Fire and Craig C. Mello were awarded Nobel Prize in 2006 for describing RNA interference. तो पता है RNA पे 3 बार नोबेल प्राइज मिल गए. RNA इंटरफेरेंस पे इनको मिला, अभी माइक्रो RNA पे अभी वाले लोगों को मिला, एंड लास्ट ईयर RNA वैक्सीन. In last 20 years only in medicine, RNAs have been able to, or the work on RNA have been able to get 3 Nobel Prize in medicine. Now we'll come to significance. अभी तक जो हमने समझा ना, वो all the things that we studied, we understood, वो हमें basic understanding में help कर रहा है. But now what we are going to study here will be something that we would directly want to write in our mains answer. Why this discovery of microRNA is significant, why this discovery of microRNA is so important, so relevant. One, it helps us better understand how our body functions, how gene get regulated. Earlier it was only transcription factor, but now this is also there. Then it has been at work for, it has also contributed in our evolution. How has it contributed in our evolution? See, initially life would have begun as a single cell, है ना? फिर an organism which have same kind of cell, but then an organism which have multiple kinds of cells. Multiple kinds of cells means in those cells different genes are getting expressed. For that, what is needed? MicroRNA would be needed, because you want to block some gene, you want to allow some gene to get expressed. So microRNA has also played an important role in evolution, and thus studying microRNA can help us understand evolution also. Then genetic research show that cells and tissues don't develop normally without microRNAs. That is, if microRNAs are not there, or if microRNAs have some issues, then your body will not be able to develop properly, or abnormal regulation by microRNA. If your microRNAs are not functioning properly, you may suffer from cancer, cardiovascular diseases, diabetes, viral infections, and other such issues, other such challenges. And therefore, when it comes to treating these diseases, can microRNA play an important role? Can microRNA based therapies, because some microRNA is not functioning properly, so some microRNA based therapies can help solve those problems. It can also be excellent biomarker. Biomarker is, it is telling if you are suffering from some disease, you say if you are suffering from dengue, you will see platelet counts reduce हो जाते हैं. That's a kind of biomarker. ऐसे ही when you are suffering from certain microRNA में deficiency की वजह से challenges, some cancer which because your microRNA is not blocking some MR, then it can clearly tell you that this microRNA is creating problems. So this presence of this microRNA or absence of this microRNA can act as biomarker, can tell you what is the issue, what is the problem that you are facing. So microRNA can also act as excellent biomarkers. For example, in cancers, the level of certain microRNA are abnormal. These create unique signature pattern that allow identification of which kind of cancer are you suffering from. So it acts as biomarker also. Do we get it? MicroRNA के advantages, significance. At the same time, what do we need to do? Development of microRNA based therapies. Already a lot of research on this is going on. ऐसा नहीं है Nobel Prize मिलने के बाद ही सबको पता लगा. Scientific community knew about microRNA and its significance in the past also, and a lot of research on this has been going on. MicroRNA based therapies, research is going on. We need to prioritize that because a lot of diseases can be solved by microRNA based therapies. Second, we should emphasize on developing suitable microRNA delivery system. Eventually, if some microRNA is not functioning properly, we'll have to deliver it within the cell, within the nucleus. उसके लिए we can use nanoparticles, we can use viral vector mechanism, we can use some other polymer for that. That thing needs to be developed. India should carry out studies on microRNA biology with the aim of producing clinically relevant molecules and diagnostics, and India should also enhance international collaboration. जैसे अभी we are collaborating in on cancer moonshoot with USA. So we need to collaborate in the field of development of microRNA to get more technologies to solve more health related challenges of India in future, जिससे हम नई therapies, नए diagnostic, नए treatment के mechanisms develop कर पाएंगे. Okay, these are certain other things that we need to do. तो in conclusion, ये एक line मैंने शायद एक ORF में article था इस about mRNA, जो Nobel Prize है उसके बारे में. They spoke about various significant aspects. तो I went through जो Nobel Prize का officially website है, मैंने उसका पूरा content read करके, हिंदू एक्सप्रेस के सारे content read करके जो सबसे relevant चीजें हो सकती है, I've included it there. But I also went through ORF. ORF में ना, that ORF was the only website where they had spoken about way forward. What are the things that India should focus upon? I like ORF a lot because वहाँ पे ना कई बार जब detailed study आएगा, they will cover a topic thoroughly, focusing on kind of कि UPSC में answers में way forward क्या होना चाहिए. ORF बहुत अच्छे से वो cover करता है. You do not have to go through it. I'll take care of it. I'll go through each and everything in ORF which is relevant here. Okay, so यहाँ पे this conclusion I've taken from there. It says that Nobel Prize for Discovery of microRNA signifies the importance of harnessing microRNA biology to develop new technologies and highlights the need for academic and clinical research to focus on translational application of microRNA biology. Basically इस बोले कि इसको Nobel Prize indicate कर रहा है कि it has a lot of potential in developing a number of new treatment and a number of new applications which can have a huge impact. It's already having a lot of impact, but its Nobel Prize indicates that it has potential to have much bigger impact on humanity. Do we get it? Do we understand it? तो फिर से एक बार सोचना कि what was this topic? This topic was about how regulation of genes happen. In our body, we have same gene in every cell, but not, we have same DNA in every cell, but in the DNA not every gene is getting expressed. Before the discovery of microRNA, our understanding what transcription factor is responsible for this regulation. Transcription factor were proteins which blocked the expression of DNA only. DNA को mRNA ही बनने नहीं देता. But then two of our scientists who got Nobel Prize this year while working on an earthworm realized that there is something called microRNA. This microRNA blocks mRNA. Gene से mRNA बन जाएगा, but mRNA will be blocked and thus gene will not be expressed. Initially when they published it, there was not a lot of significance given to it because it was considered a peculiarity of earthworm but later they showed that microRNA exist in complex living organisms also, including humans. In humans, around 1000 microRNA have already been discovered, and thus its significance is immense in understanding regulation of gene expression in human beings, in understanding our evolution, in dealing with various diseases which may occur because of poor functioning of mRNA, and in developing future treatment also. Then it also plays an important role as a biomarker. Diagnostic maybe microRNA can play an important role. So what do we need to do? We need to develop microRNA based therapies. Already a lot of research and development is happening. That needs to be prioritized. We need to develop tools through which microRNA can be delivered. इंडिया रिसेंटली बायोटेक्नोलॉजी के लिए बहुत सारे इनिशिएटिव्स लेके आया है, उसमें माइक्रो आरएनए बेस्ड इनिशिएटिव्स हैव टू बी प्रायोरिटाइज्ड, एंड इंडिया शुड आल्सो फोकस ऑन इंटरनेशनल कोलैबोरेशन इन दिस फील्ड. हम we have several initiatives under which we are collaborating with different countries in the field of biotechnology. Within biotechnology, microRNA based collaboration is also something that we need to prioritize. This Nobel Prize indicates the significance that microRNA has for benefit of humankind, whether in the form of new therapies, whether in the form of overall diagnostic and improvement of human life. This is the overall summary about the topic. आपको मेंस में यही सोचना पड़ेगा, बट जब प्रीलिम्स के बारे में प्रीलिम्स में पढ़ने जाओगे ना, तो थोड़ा ध्यान से अंदर की चीजें भी देखना, बिकॉज sometimes they go into, जैसे RNA interference technology, no one had ever expected that UPSC will ask question on RNA interference technology जब वो आया, right? But now when you see कि RNA interference technology पे यार नोबेल प्राइज मिला हुआ है, इतना तो पढ़ना चाहिए था. अब इन द सेम वे माइक्रो आरएनए टेक्नोलॉजी पे, अब नो वन वाज एक्सपेक्टिंग कि माइक्रो सैटेलाइट मिनी सैटेलाइट पे क्वेश्चन आएगा, दो वी वर कवरिंग दोज इन क्लासेस, बट दे आस्क्ड क्वेश्चन ऑन मिनी सैटेलाइट माइक्रो सैटेलाइट्स. अभी रिसेंटली उन्होंने क्वेश्चन पूछे थे ऐसे ही, इफ दे आस्क क्वेश्चन ऑन माइक्रो आरएनए, वी शुड नॉट बी लाइक ये कुछ बहुत कॉम्प्लेक्स आ गया. एंड मोर ओवर, समझो इस बात को, नोबेल प्राइज हैज बीन गिवन ऑन दिस इशू, एंड इफ वी आर एबल टू अंडरस्टैंड दैट, Nobel Prize is given on what? Nobel Prize is given on the biggest advancements which are impacting humankind. और हम वो even if we are not from science and technology background are able to understand, so that's a achievement for us. That shows कि हमारा overall understanding improve हो रहा है. And for those of you who may have found many of these terms very complex, don't worry. जनवरी में जब हमारा टारगेट प्रीलिम्स फेज स्टार्ट होगा, वी विल बी स्टार्टिंग विद साइंस एंड टेक्नोलॉजी, एंड देयर बिफोर जंपिंग ऑन टू दिस टॉपिक, मैं वहां पे ये टॉपिक दोबारा रिवाइज करवाऊंगा आपको प्रीलिम्स के टाइम, बस प्रीलिम्स के पर्सपेक्टिव से, वे फॉरवर्ड और वो सब नहीं कराएंगे. बट वहां पे बिफोर टीचिंग दिस, आई स्पेंड 1-2 आवर ऑन द बेसिक्स ऑफ बायोटेक्नोलॉजी. आई स्पेंड अ लॉट ऑफ टाइम. मैंने बोला कुछ सब्जेक्ट, साइंस, एनवायरमेंट, इंटरनेशनल बॉडीज, मैपिंग, international relations. For these things you will not be referring to any source. I'll make sure that each and everything in the most updated way is there in your notes only. आपको हिस्ट्री, ज्योग्राफी, पॉलिटी, इकोनॉमी के बेसिक्स और एथिक्स, बिकॉज मेंस का भी प्रिपरेशन कर रहे हैं, आपके टेस्ट के सिलेबस में एथिक्स भी रहेगा, है ना. विल मेक श्योर दैट by December, January you're revising ethics also 2-3 times other than revising these things. तो ये हिस्ट्री, ज्योग्राफी, पॉलिटी, इकोनॉमी के बेसिक्स, करंट अफेयर्स नहीं, करंट अफेयर्स में कराऊंगा, इकोनॉमिक सर्वे ऑफ इंडिया, बजट में कराऊंगा, मेंस के बुलेट पॉइंट का एक सिलेबस में करा के दूंगा. बेसिक्स ऑफ हिस्ट्री, ज्योग्राफी, पॉलिटी, इकोनॉमी, यू शुड नो. एथिक्स यू हैव टू डू इट योरसेल्फ. आई विल कवर सम टॉपिक्स, जैसे अब लाइफ ऑफ रतन टाटा हमें क्या क्या एथिकल वैल्यूज दे सकते हैं, क्या क्या लेसन्स दे सकते हैं, उस तरह की चीजें हम करेंगे, बट नॉट, क्योंकि इट्स मोर ऑफ अ वेरी स्टैटिक टॉपिक. हम करंट अफेयर्स में डू नॉट एम्फेसाइज अ लॉट ऑन दैट. तो आई होप ये टॉपिक माइक्रो आरएनए इज अंडरस्टूड बाय ईच वन ऑफ अस. वी आर ऑल कंफर्टेबल विद इट. ओके, नाउ विल गो टू द नेक्स्ट वन. लेट मी टेक दिस ऑनलाइन डाउट्स जरा जल्दी से. Will you cover GS2 before December like you covering topics of agriculture and disaster management GS3? नहीं भाई, I'm not promising that your current affairs of everything GS2 will be completed by this before December. Before your prelims, all your GS2 things would be completed before your mains. मैंने बोला सिलेबस का एक बुलेट पॉइंट कवर करना, वो प्राइमरली हम टारगेट मेंस फेज में करते हैं, जून जुलाई वाले पीरियड में, है ना. आई एम नॉट प्रॉमिसिंग इट कि आपका पूरा GS2 और पूरा GS3 कवर कब हो जाएगा, अभी दिसंबर में हो जाएगा, बट आई विल ट्राई टू फिनिश एज मेनी टॉपिक्स एज पॉसिबल सो दैट वहां पे यू हैव लेस लोड. जैसे दिस मंथ आफ्टर कवरिंग 3-4 बुकलेट्स फॉर व्हाट इज हैपनिंग इन द करंट अफेयर्स, आई विल जंप ऑन टू इंफ्रास्ट्रक्चर. मैं आपको 1-2 बुकलेट दूंगा पूरा इंफ्रास्ट्रक्चर पे, बेसिक्स से. मैंने आपको क्या बोला, बेसिक्स ऑफ इकोनॉमी आना चाहिए. आई डू नॉट वांट यू टू नो एग्रीकल्चर भी, इंफ्रास्ट्रक्चर भी, लैंड रिफॉर्म्स भी, जो सब सिलेबस में, वो करवाने की जिम्मेदारी हमारी है. वी विल कवर दैट इन क्लास.